so great to be here today, and it's actually kind of great to also be last, especially after so many people whose work I've read for so many years and admire, and thank you both for inviting me so much. It's such a humbling experience, and it really, when I was preparing this talk, it brought to mind a quote by a very famous scholar, that is Google scholar, that reminds me every time that I run a search that I stand on the shoulders of giants. And just to be accurate, of course, this is not Google scholar, this is Isaac Newton, but it's still true. If I've seen further, it is because I stand on the shoulders of giants, and to all of these giants I owe an extreme debt of gratitude, which really leads me into where I like to start every talk, which is all the people who've contributed to this work. This is a team at Columbia University where I did my graduate work, my advisors and my collaborators and students there, my colleagues at Pittsburgh and Carnegie Mellon. I'm going to show you some data that we collected together. At Yale, my lab, and my mentors, especially Kathy Carroll. I definitely wouldn't be here if it weren't for you. And of course, without funding sources, we also wouldn't be here. So thank you particularly to NIDA for funding a lot of the work that I'm going to show you today. I'm going to try to make a few, hopefully not too complicated points in my talk. I'm going to first argue that craving is a key process in addiction. It used to be much more controversial. Some of you might still want to argue with me. I'm going to talk about how the ability to regulate craving is really a key process in addiction and abstinence as well. And I'm going to suggest that both of these are really potential biomarkers that we might want to consider for addiction and for treatment. And then I'm going to introduce you to the ROC Task, which is a task that we designed and built originally to understand what the neural mechanisms are underlying regulation, to test the efficacy of strategies in an experimental way. We are now looking at using this task to differentiate between different kinds of strategies and how effective they are for different people, and what the neural systems that they might recruit are, and to predict outcome using individual difference approaches, to track change over time, and to train regulation. And if you think this is an ambitious list, trust me, I meant to show you much more data than this. So the word craving kind of floated in the air in this conference by many, many people, but because I'm going to spend the next 20 minutes talking just about it, I figured it's good to kind of keep us all in the same page, or maybe keep you all on my page. So most people, when they think about craving, they think about it as a strong desire or kind of a feeling of wanting. And what is maybe important to keep in mind is that this has been a concept that has really been investigated by philosophers for thousands of years now. So the first was, of course, Buddha, the Buddhist philosopher. He said that craving was the cause of all suffering. Spinoza said that craving is the very essence of man. Bertrand Russell said that it prompts all human activity. And it's maybe nowhere more apparent than around drugs, food, and behaviors like gambling, or maybe for some of you, Facebook or checking your email. But the idea is that really craving is really common, and it's maybe something that, at least according to epidemiological studies in the context of food, 100% of people report experiencing, according to some estimates, experiencing craving for a specific food at least at some point in their lives. So really, all of you should have some kind of intuitive sense of what it is, and just in case you don't, I figured I'll bring these lovely chocolate croissants here for you to stare at for a moment. You can maybe think about how fresh they are, and maybe they're chocolate croissants, and so there's molten chocolate inside. Maybe some of you might want to have your croissants with some hot coffee. This is looks like pretty good cappuccino. And so, you might, if you didn't expect, I see some of you smiling, so you're probably feeling it. There's a commercial I really like to show, but it's for afternoon food, not for morning food. And so the experience itself, it's really common, and you probably have a sense of it, but it's not really quite simple. It's actually a multidimensional construct that includes a couple of components. There's traditionally thought of as an affective component, that feeling of wanting. There's a cognitive component, which is both the thought that you want something, but also expectancies. If I have this, I'll feel better. If I don't have it, I'll feel worse. And of course, there's a motivational component, which is associated with the impetus to act on these thoughts and feelings and kind of consume that thing which you crave. Together, many people have considered these processes, or this multidimensional construct, as a key process in addiction, which has culminated in it becoming recently a diagnostic criterion in DSM-5 for addiction. Now, it becoming a diagnostic criterion really rests on now several decades of data that show that drug users typically report craving as one of the sources or the causes for relapse. Much data correlates drug use and craving, both retrospectively, prospectively in an EMA study, so kind kind of in a more moment by moment tracking way. And there's also quite a few studies that have shown that craving actually predicts, in formal prediction models, predicts drug use and relapse. And this is again with various kinds of methodologies, including EMA, across various substances, from food to nicotine to stimulants and heroin, and also across time resolution. So predicting both immediate drug use, but also sometimes relapse a few weeks in the future. And when I talk about craving and outcome, I don't want you to get the wrong impression that I think that craving is the only thing that predicts outcome. Of course, addiction is a very complex disorder, and drug taking is a complex behavior. But what I am trying to say is that craving consistently predicts a significant proportion of the variance of drug taking. Tiffany's group last year, led by one of his graduate students, I think, have published a meta-analysis for the lab studies of smoking. So these are not clinical studies. These are just studies in which people come into the lab, they report some form of craving, sometimes cue-induced or not, and then in the lab have an opportunity to either work for cigarettes or actually smoke. And in this meta-analysis, so again, only lab studies, does not include the clinical data, they found, depending on the analysis, a prospective aggregated effect size between 0.18 or 0.34. And this is, medium, small to medium effect sizes, still predicting a significant portion of the variance in smoking outcomes. We took a slightly broader approach. My graduate student and I just submitted this paper with food, and we looked across all of the food, weight, and immediate consumption studies. And so we again did this kind of effect size meta-analysis. There's about 50 different statistics in this meta-analysis. So in all of these studies, again, people came into the lab, reported some kind of cue reactivity or craving, and then prospectively their eating was measured, or their weight or BMI was measured as far as 2 years after that session. And what we find is a combined medium or medium to large effect size. For cue-induced craving, we found 0.35 aggregated effect size, for tonic craving of 0.3. And I also wanted to put in this analysis we did just for imaging cue reactivity and its prospective effect on weight or eating. And while you probably all know that imaging is sometimes associated with puzzlingly high correlations, it is interesting to see that the effect size here is a little bit bigger. We're also now doing the same thing for across all of the nicotine studies, both lab and clinical and other kinds of drugs. We don't have them all entered, but we have quite a bit of them, and we're actually finding a larger effect size when we include the clinical studies in a meta-analysis. And so while again this suggests maybe about 10 to 15% of the variance being accounted for by craving alone, and it means that there's still a lot of the variance is accounted by other factors. I hope that we can agree before we move forward that craving is at least an important contributory factor to drug use and relapse. So what's next? We're going to kind of look under the hood. When I was a kid, I really liked to take things apart when I thought they were cool and interesting, and craving is cool and interesting, so I like to take it apart. And so we really wanted to try to understand what the neural mechanisms of craving might be. And this is where we get to be translational and hip, according to Kent, because there's really a rich animal literature that has already looked at the neural mechanisms of craving. It's typically found that cue conditioning specifically, or even in the animal literature they call cue-induced craving, is a conditioned process that's easily done in the lab, and actually even in humans, even after a single conditioning session with chocolate, there's now data that they become conditioned to the cue with craving. We know that it involves dopamine, and we know also that there's an involvement of the ventral striatum or the nucleus accumbens in animals. But the emerging human literature is really not as consistent as we might hope. So there are some regions that come up more than others, but really some regions are actually being hotly debated. So the involvement of the insula, for example, some people think very intimately involved, some people think less so. And between 1996 and 2012, there's about 50 neuroimaging studies of cue-induced craving that have been done, and they vary across drugs, paradigms, cue types, imaging procedures, and of course analysis procedures. And so really there isn't what I think at least of as kind of the representative regions that we know are implicated. And so for that we did a meta-analysis of imaging studies, and so far we have all the papers through 2012. We're still working on 2013 and 2014, and we entered them all into a database. We've done this before, and you can see there's quite a bit of variability in the kinds of stimuli that have been used, by the drugs that have been used, and of course the imaging methodology. And so what we're really looking for are voxels in the brain that are most consistently active across all of these studies. This is a kind of a method slide. I'm not going to belabor it too much, but the idea is that we really enter for every study the voxels that have been reported as significantly active, more in the drug cue condition than the non-drug cue condition. We convolve them with a little kernel over here, and then we weigh them by the number of participants who contributed to the analysis, and then perform Monte Carlo simulation and end up with this heat map. And we've published on these methods a couple of times already. And when we look across these 42 studies of craving, what we find is what we can now maybe think of as maybe the closest neuroimaging biomarker to where craving might live in the brain. We're actually using some classification analysis with these data as well. And you see the ventral striatum quite robustly and bilaterally, the anterior cingulate cortex extending into the medial prefrontal cortex and the orbital frontal cortex, the amygdala bilaterally, the thalamus, and of course the VTA and the substantia nigra, these dopamine-producing regions. And when we look at this network of regions, clearly, because this is what the analysis was, we can say that these are the regions that are consistently activated during cue exposure and craving. But it's important to note, and maybe you want to ask me about that later, I have lots of data here, but I didn't have time to show, there are actually between drug differences, and there are also differences with food and with gambling, some overlap, but not complete. And some people might look at this and say, oh, look, all of these regions are along the dopamine pathway, of course, owing to the fact that this is a conditioned or a learning-based process. Some people might look at this and say, look, they're default mode regions. What you might also notice is that these are regions that are commonly activated in emotion. This is a slice from an emotion meta-analysis that we published some years ago across 168 studies of emotion. And specifically, you see that these are regions that overlap with both negative and positive emotion, which might also make some intuitive sense when we think about craving. Either way, if these are the regions that are consistently activated by craving, these would also be our targets for modulation by regulation strategies, which is the next part of this talk. So addiction has often been formulated as a regulatory failure, and many of the models of addiction, people talk about it in this way, but you really need to look no further than DSM-5, where some of the diagnostic criteria really inherently define it as a regulation of failure. So using larger amounts and for longer than intended, continued use despite knowledge of ill consequences, and desire to stop but failing are all kind of examples of how the disorder is really defined by not being able to stop yourself from behaving. On the other hand, we know that when people apply regulation of craving, it does decrease use. So for example, cognitive and relapse prevention methods are effective in reducing substance use. And as Kathy talked about yesterday, there's really a focus on identifying and avoiding drug use, high-risk situations, and training and coping strategies, including coping with craving. We know that acquisition of cognitive strategies during CBT is associated with reduced relapse. This is work by Kathy and Brian Killick. And of course, application of cognitive strategies induces craving and relapse, and this is really elegant work that has used perspective and EMA methods to actually check people in moments of craving, and when they kind of engage cognitive strategies, they actually, this is with smokers, they smoke less. And so as such, regulation of craving is really a key component in abstinence and kind of really important when we think about treatment. And so to really study that in a laboratory setting, we came up with a ROC Task. We initially designed it to directly measure both the causal effect of regulation strategies on craving and also the neural mechanisms of both craving and regulation kind of in the same setting. The time course of this task, it looks exactly like an emotion regulation task. People see a fixation cross, they get an instruction, and then they see an appetitive stimulus. We often show them food as a control stimulus, and then if it's cigarette smokers, they see cigarettes. We've done this now with methamphetamine users, cocaine users, and we now have work with Godfrey with individuals who are bariatric patients, and I have a graduate student who works with binge eating disorder. They see only food. They see another fixation cross, and then they report their craving on a trial-by-trial basis. In the first set of studies, we used cigarette smokers. They saw food and cigarettes. And then they got two of these instructions that really orient them to think about the pictures that they're about to see in particular ways, and they're trained to use this prior to the experiment. The first instruction is really modeled after what drug users typically do automatically when they see drug cues. They focus on their immediate feelings and the positive immediate feelings particularly. So we tell them specifically, if you were shown a picture of a cigarette, you should think about how smoking will make you feel when you light up. Focus on the smell and how smoking the cigarette will taste. And the cue word we use in the scanner is the word now. The second instruction, taken straight out of CBT manuals, orients participants to think about the long-term negative consequences of using, or at least in smokers, it's long-term. We now use both long and short-term consequences. And we tell them if you were shown a picture of a cigarette, you should think about the consequences smoking will have for your lungs, for your heart, and for your likelihood of developing cancer. And the keyword we use for this is later. So again, this is what they see, the instruction word now or later. They see either cigarettes or food. So first, what happens to self-reported craving? We actually, when we first tried to publish this work, people gave us a lot of difficulty, but this is now, I think, slightly more established. What you see here, red is the now condition, blue is the later condition. Both with food and with cigarettes, we see a significant reduction in self-reported craving. And I should mention that this has now been replicated quite a bit. This is with smokers, cigarette smokers. This is with cocaine. This is a paper we have with methamphetamine users under review. Nazir Naqvi and John Morgenstern published this paper with alcohol, and this is with food, including our work, which is not yet published. And when we look at the imaging data, what we expect to see when we contrast now and later is, of course, regions that are associated with craving, because that's a craving condition, and we see exactly that, the orbital frontal cortex, the anterior cingulate, the VTA, the subgenual cingulate, and the ventral striatum, which you can imagine map quite well to the meta-analytic findings that I showed you just a couple of minutes ago. And when we contrast the later condition with the now condition, what we would really expect to see here is something that's related to this regulatory ability, and indeed we see none of that activation in the craving condition. Instead, we see recruitment of what are considered to be classic cognitive control regions, like the dorsomedial prefrontal cortex, the dorsolateral, and the ventrolateral prefrontal cortex. Now, just to put this in the context of kind of the larger emotion regulation work, much emotion regulation work has used negative stimuli and used reappraisal to negative regulate response to these stimuli. We recently published a meta-analysis with Kevin Oxner. This is work that I started in grad school and was continued by Jason Buell and Jen Silvers. And this is a meta-analysis of all of the regulation studies using reappraisal. And you see that really in our study, the results map pretty well. So they're kind of really recruiting regions that are commonly activated in reappraisal or emotion regulation. We also took an individual difference approach and looked for regions that really tracked with people's experience of craving. And what we see here is that the ventral striatum positively tracks with craving, such that the more craving people reported, the more activity in the ventral striatum they exhibited as well. And we see the exact reverse in the dorsolateral prefrontal cortex, such that the more they recruit this region, actually the less craving they report. Now, suggesting that it also tracks with craving just in the opposite direction. There's also mediation analysis, and I'm not going to show you. What I am going to show you is that this has now been replicated by both Neurovolcal with PET and by us with fMRI. This is the slide that Kathy flashed at you yesterday briefly. So this is, again, this negative correlation, the dorsolateral prefrontal cortex with self-reported craving in this task. So I showed you that we're using this task to measure craving and cognitive regulation, both in the kind of a main effect and individual differences related to recruitment of regulatory regions. But we can also use it to compare the efficacy of strategies related to drug-taking behavior, assess treatment-related change, and train regulation itself. And I'm going to start with strategies, and I'm going to focus on mindfulness. As a long-term meditator, this is kind of my I guess, disclaimer here. I care about this a lot. And Judd gave the John Kabat-Zinn definition yesterday. It's moment-to-moment, non-judgmental awareness of what is, right? So it's the ability to be here right now, notice my feet on the floor, maybe notice that I'm a little bit nervous still talking to all of you, but just letting it be exactly how it is. And I really like this definition because it kind of highlights the two components of mindfulness, which is attention to the present moment, but also acceptance of whatever arises in the moment. And while mindfulness is not running around on the beach holding hands and singing Kumbaya, as Judd said yesterday, it might look something like this. So you sit in front of the sunset and meditate. This is actually me. And so with respect to mindfulness, we asked, is mindfulness a good strategy to regulate craving? And is it different from CBT-based regulation? We use the same kind of paradigm. This is 43 treatment-seeking smokers. But we did give them these new instructions. And we just switched the regulation instruction to noticing and accepting without judgment whatever arises. And we specifically told them, if you see a cigarette, notice any thoughts, sensations, and craving that might arise. You can ask yourself, can I just be okay with this feeling? Simply notice and accept any sensations that you experience. And the keyword we used for this in the scanner was the word accept. The trial time course looked exactly the same. These are the two instructions, feel or accept. And when we look at first self-report, we see again a significant reduction in self-reported craving. I'm not going to talk about individual differences in the ability to do that, but there are. And then we also see a reduction in activity in the subgenual cingulate and the ventral striatum, as we saw in the previous study. What we're not seeing here, however, is any recruitment of prefrontal cortex. In the paper, which if you want, you are welcome to read, we kind of go after finding the PFC in a few different ways. We are not observing any recruitment of the prefrontal cortex with this mindfulness-based strategy. So it suggests that while mindfulness-based regulation is effective in reducing craving and reducing craving-related neural activity, it really relies on different neuromechanisms than CBT-based regulation of craving. And I have an elaborate set of theories here about mindfulness being related to a kind of reduction in reactivity instead of increased regulation, which I'm happy to talk about. We also have data now with thermal pain that is very, very consistent with this. So just briefly at the end of my talk, I'm going to ask the question if we can train regulation of craving. Now we're using this task also as a kind of a probe for increase in regulation skills in the context of CBT with Kathy and Brian's study, as she talked about yesterday. But we also kind of weaponized it as a treatment on its own, or at least a treatment in formation or in testing. And so the idea here is that people can actually use the ROC task to practice the regulation in the presence of cues over and over and over. So they really do the task, except really 75% instead of 50% of the trials are regulation trials, so they really get to practice it over and over. The conditions that we've included so far is a no training condition, a CBT-based training condition, so doing the later strategy over and over, or doing the accept strategy over and over in the mindfulness-based training. And we've now have 3 studies that we've conducted, kind of increasing the dose now from 30 minutes to 60 minutes to 360 minutes, or 6-hour sessions to test this. So in the first 2 studies, they came to a single lab session that was either 30 or 60 minutes, and we only did the CBT training or the no control training. So in the CBT-based training, they really just practiced over and over, thinking about the negative consequences of smoking every time they saw a cue, over and over and over. And then we followed them up to a month. And just to quickly show you the results, what you see here, and this is from 60 subjects, is that compared to the control condition that received no training, even out to a month after a single session, this is with 60 minutes, the CBT ROC training was actually associated with 37% reduction in number of cigarettes per day. So I'm not showing you kind of full abstinence, but people are really significantly reducing their smoking even with an hour of training, which is really exciting. What we're also seeing is that the amount of craving that they're reporting in the last few trials of the training actually predicts their smoking at follow-up. So those individuals who report the kind of lowest craving at the end of training are the ones who also smoke the least a month out. We just got funded as a part of the Psychotherapy Development Center to run this randomized controlled trial where they're receiving 6 laboratory sessions of this training over the course of just a few weeks. We have now follow-up extending up to 2 months, and we've of course also introduced the ROC task and fMRI both pre and post training. And here we're comparing specifically CBT and mindfulness-based training, and I'm really just going to show you preliminary results. This is from just the pilot subjects, or 12 in the sample, just to kind of give you a taste. You know, I kind of can't help my excitement because it's really interesting and consistent with some of the insights that I think we've started to glean about how mindfulness and CBT might be different. So this is change in reported craving during the scanning session from pre to post training, and I want to orient you to, this is the MBT training in the accept condition and the CBT training in the later condition, and what you see is that both groups in their strategy condition really reduce craving significantly from pre to post training. But what you see here is that only people in the mindfulness-based training condition reduce their craving even in the craving condition. So they're just showing in general reduction in reactivity to these cues that are being presented to them. And again, this is consistent with this idea of reduced reactivity rather than increased regulation as a result of mindfulness training. Now again, preliminary data, but this is the imaging data, and what you see here is that for individuals in the CBT condition, from post-training to pre, they show an increase of recruitment in the dorsolateral prefrontal cortex during the regulation condition, whereas we don't see that for the mindfulness-based training group. All right, so I set these lofty goals. I got through them just in time. Craving is a key process. The ability to regulate it is really important for abstinence, and so we really think that if we're able to track these processes and their neural correlates along treatment, we can probably have I shouldn't say probably, we maybe will have better probes for understanding how they change over time. I especially am interested in the way that mindfulness might offer a particularly non-PFC dependent yet robust way for people to regulate their craving, and the degree to which this might be maybe particularly suitable for some subset of participants for whom recruitment of prefrontal cortex might be more difficult, potentially even impossible. And I hope that you stay tuned. We should have a lot of data in about 4 years, and thank you all for listening.